All right, on to 19.3. So talking a bit about cancer, which will also be revisited in Chapter 12 a bit, because cancer is largely tied to the cell cycle and cell cycle regulation. So here we're going to talk about regulation before the cycle itself in the next unit. So gene regulation is basically when it goes wrong, depending on which genes are overexpressed or underexpressed. One of the big results could be cancer. And cancer is essentially a mistake or error in the cell reproduction process. So a tumor is a giant ball of the same cell, many copies of the same cell. And if you're making many copies of the same cell, there's an issue in regulation. So the genes that normally regulate cell growth and division, whether they be for growth factors, the receptors that we showed you some earlier, activators and so on, or various intracellular molecules of pathways, all these can be messed with to cause cancer. So genes that cause cancer are called oncogenes. Essentially that means genes that promote the cell cycle to go overboard. We call those oncogenes. And proto-oncogenes are normal cellular genes that code for cell growth or cell division. And those proto-oncogenes are the healthy ones that can be, through mutation typically, they can become oncogenes and thus cause cancer. Oncogenes are cancer-causing genes. So here we take a proto-oncogene and make it excessively active such that it becomes an oncogene. Here's the proto-oncogene that you see there. Normal protein in slight excess. And if you amplify that gene and make many copies, you're going to make many more copies of that, potentially causing more cell division as well. Excuse me. And if you mutate that proto-oncogene and essentially mess with its control element, so eliminate the cell's ability to down-regulate this proto-oncogene's expression, as you see here in this control element mutation, that's going to cause this gene to become an oncogene, and now you're just going to make tons and tons of this protein, and this is going to keep going and going and going. This protein enhances the cell cycle or causes the cell cycle to happen, or in layman's terms, it makes the cell divide, if you like. Now, on the opposite end, we have tumor suppressor genes, and these are genes that code for proteins that inhibit cell division. They block abnormal cell division. And so there's many proto-oncogenes and tumor suppressor genes that that encode proteins that inhibit pathways and stimulate the pathways respectively, and they sort of fight against one another. It's always an interplay or a balance between the two as to when to divide and so on. So here's an example of one. This is the RAS protein encoded by the RAS gene. And the RAS protein is what's called a cyclin. It's a protein that is associated with the cell cycle. And here we see, this looks hopefully familiar to you, where we have the dimerization, and here is actually this tyrosine kinase is associated with this G protein, and the G protein is RAS. Now G protein, here a mutation happens that caused the RAS to be hyperactive, and thus now the RAS protein is the product of the RAS oncogene, which triggers the cell cycle. Here you see transcription factors and RNA polymerase and so on to bind to the DNA and to, not RNA polymerase, sorry, bind to the DNA and to copy. This would be DNA polymerase among other proteins. Or it could be, as I mentioned, RNA polymerase that makes RNA, transcribes it into a protein, and this protein stimulates the cell cycle. So a variety of different ways, but essentially we have a mutation that triggers an overreactive or overactive protein that causes the cell to divide over and over. RAS is one very common one that can get mutated. So you see that here described. On the other hand, we have P53. P53 is a tumor suppressor gene which codes for the P53 protein. P53 protein under normal circumstances is good. It's a tumor suppressor protein that blocks the cell cycle from occurring. It inhibits the cell cycle. But when the P53 protein gets mutated, it cannot activate transcription and thus cannot inhibit the cell cycle because it doesn't actively or properly transcribe the DNA. P53 mutations have been associated with somewhere around 70% of cancers. So not all cancers are tied to p53, but it's very, very highly studied because it's found in such a large variety and amount of, or number of, cancers. So here we're showing you a mutation that knocks out the p53 gene, and as a result you can't turn off the cell cycle. Normally p53 turns off. It's a tumor suppressor gene. It downregulates or slows the cell cycle. p53 doesn't work, so it leads to excess cell division. Now, normal cells can be converted to cancer cells through multiple mutations, which directly affect proto-oncogenes and tumor suppressor genes. So the proto-oncogenes would be converted into oncogenes, and the tumor suppressor genes, as we saw with p53, would be turned off and blocked from their ability. And thus, cell division would occur rapidly, abnormally, and cause the formation of a tumor or cancer. So anything that can cause mutations can essentially cause cancer, and we call those things either carcinogens in the environment or mutagens, whether it be UV rays from the sun or eating burnt toast. The black stuff would be a carcinogen, and so on. Lots of different things in our lives are classified as carcinogens, and you want to minimize, of course, your consumption of carcinogens. So here's an example of cancer-forming normal epithelial cells of the colon, of the large intestine. We have a small mutation that causes an abnormal cell division. We get a small accumulation here. This is small benign growth, but as this continues, if we have an additional mutation potentially, and thus activation of an oncogene here, that continues to grow even bigger, and now it's an even larger growth. It's still benign, benign meaning it doesn't spread through the bloodstream. And now we have this final enlargement that you see here, loss of tumor suppressor gene p53, and now it's become malignant. So cancer is not the result of one mutation. Cancer or tumor is the result of many successive mutations in the correct places that cause the formation of this malignant tumor, and you notice it's going to try and spread through the bloodstream there. Now, we do know that certain viruses can cause cancer by altering or mutating the cell's DNA. One example would be HPV. So many young women listening to this might have been vaccinated against HPV, or human papillomavirus, which is believed to be tied to some female cancers, ovarian cancer, uterine cancer, along those lines. So if you can block infection by this HPV, then potentially you could inhibit or minimize the likelihood of cancer. HPV is a sexually transmitted pathogen. The effect in men is unknown, if any, but the effect in women is known, and thus the vaccination. In addition, you can inherit it, of course, genetically. If a mutation occurs in mom to cause the formation of an oncogene, so a proto-oncogene is converted into an oncogene, or, of course, to the tumor suppressor gene, then when that mom passes on that gene to her offspring, that offspring would have an increased likelihood of cancer. So cancer has of course, genetic component. And keep in mind that the mutation in the mother, if the mutation occurred in a cell of the mother's eye, it wouldn't make a difference to the baby because that cell is not part of the offspring, the egg formation. But if it occurs in the ovary, and specifically the germ cells that will become the eggs of for the mom that is going to be the baby, well then, of course, that mutation is going to be passed off to the child. So some examples of this are BRCA1 and 2. This stands for breast cancer 1 and breast cancer 2. These are genes where if you carry one BRCA1, the mutated form, which is called BRCA1, the normal form is not called BRCA1. It's normal in the majority of the population. If you carry BRCA1 by itself, or you carry BRCA1, as it's called, BRCA2, by itself, I think your incidence of getting breast cancer is heightened by some 50%. If you carry both genes, your likelihood of getting breast cancer is somewhere close to 100%. So some women are choosing to get tested for both of these genes, and if they carry both of them, they are being sometimes proactive in having double mastectomies to remove their breast tissue early to eliminate the possibility of having breast cancer, and then having breast augmentation to replace it, essentially. But that's a big question that some women have to weigh, and could potentially be life-altering, of course, right? So the majority of DNA we know is not genes, and we used to call this junk DNA. We no longer call this junk DNA, of course, because we know it's enhancers and other regions, other important components, introns and so on, that are important in expression of genes, not just the information itself, but turning things on and off. They play important roles. So if we compare eukaryotic and prokaryotic genomes, well, eukaryotic genomes are much, much larger, which is why, of course, we can't take eukaryotic DNA in its entirety from one cell and put it into a prokaryote. It just wouldn't fit. The genes themselves of eukaryotes are much, much longer. And there's also far more non-coding DNA between the genes than you find in prokaryotes. And here you see sort of a comparison here in a human genome. The eukaryotic genome, 98.5% do not code for proteins, rRNAs or tRNAs. They code for other things, such as found in introns or repetitive sequences like polyase or various transposons and so on. What exactly are transposons? I just said that word. Well, Barbara McClintock, who did experiments on corn, with Indian corn, these beautiful different colors, she was the first one to cite wandering DNA, as was referred to, and which we now call transposons. So eukaryotic transposable elements come in two forms, either transposons or retrotransposons. So basically transposons are when DNA gets copied, and then this copy of just this one DNA fragment gets reinserted somewhere else in the genome. Not entirely clear why this occurs, but this occurs on a fairly regular basis, and as you might imagine, that increases the size of this eukaryote's genome. So historically or ancestrally, if we look back at the size of the genomes of different organisms that are related in a family tree, we see that more present-day organisms have larger genomes, because in part due to transposons. There's also retrotransposons. This is more the copy-paste, where this DNA gets converted into RNA, and then reverse transcriptase, like we saw in HIV or other retroviruses, converts that RNA back into DNA, and that double-stranded DNA will now get reinserted into the genome as a retrotransposon. Again, increasing the size of the genome or potentially making a second copy of a gene. So now there's two copies and increasing the amount of protein that's made. So multiple copies of transposable elements and sequences that are related are found all throughout the eukaryotic genome, all over the place. In humans and other primates, there's a large portion of the transposable element. Related DNA consists of a family of similar sequences called ALU elements. So here we'll talk about ALU elements in humans, and this is just copied directly from Wikipedia from when this lecture was written. But it's a fairly well written thing, so I'll just read it off as you read yourself. Most human ALU sequence insertions can be found in the corresponding positions in the genomes of other primates. So these transposable elements are found in the same places on the same chromosomes in other primates. However, there's about 7,000 ALU insertions, or short sequences, that are unique to humans. Now, these ALU sequences in primates, they form a fossil record that we can look at them and use them to track ancestry and similarities. So, form a fossil record that is relatively easy to decipher because ALU sequence insertion events have a characteristic signature that is both easy to read and faithfully recorded in the genome from generation to generation. The study of ALU sequences thus reveals details of ancestry because individuals will only share a particular ALU sequence insertion event or insertion if they have a common sexual ancestor. So ALU insertions are not the only type of DNA that's found repeated. There's simple sequence DNA where you can have many copies of tandemly repeated short sequences that are found in the DNA, not always known what their purpose is. But we often find these repeats in centromeres, the DNA that codes for proteins involved with centromeres, and also in the telomeres, the ends of the chromosome, where they both play structural roles. Not entirely clear why there's the repetitive sequences. In telomeres, we might know more so than in centromeres. So most eukaryotic genes, there's one copy per haploid set of chromosomes, so that's why on a homologous pair we have an A and an A. But quite often, we find that there's more than one A on a chromosome. There might be an A and an A and an A. And this we call a multigene family, where there's collections of identical or very similar genes found throughout the genome and often on the same chromosome. So here we see an example of a multigene family where there's many genes associated with one protein. So each different gene codes for a polypeptide that all of them combined together in the quaternary structure makes one big protein. That's one example. Another example of a multigene family here, we have the globin family of proteins. Globins are believed to have been originally a transposon that got reinserted back into the genome many times and over many years got mutated to form different types of proteins. So here we see the alpha globin gene family on chromosome 16 and the beta globin gene family on chromosome 11. They're all globin proteins, all very, very similar, but code for different proteins here, of course. But they all originated, as you might imagine, from one original globin protein. Now, how did they get different? Well, mutations, of course. Mutations of DNA contribute to genome evolution that we can see. This could be an accident in cell division, where an extra copy of one little part or even all of the genome can be added, and these different mutations or errors can accumulate. So here, for example, we have unequal crossing over, where instead of exchanging equal sized legs of DNA, they exchange different sizes. So one chromosome has a duplication, and one chromosome has a deletion, both of which will cause potential problems, both of which are frame shift mutations. Here we see that one ancestral globin gene, it's a transposon, it gets duplicated, reinserted. Now we have two mutations over time, causes these two genes to be different slightly. So this is alpha globin, this is beta globin, and all these that subsequently get reinserted and then mutations occur make up this alpha globin family, all on chromosome 16. And then you might imagine the same scenario for the beta globin on chromosome 11. So there's some examples of transposons there. And so these globin genes, you might know hemoglobin, which carries oxygen in the bloodstream, is derived from the original globin protein, or immunoglobulins. That's another word for antibodies. Antibodies are globin proteins from the ancestral globin. Here you can see amino acid sequence similarities between alpha globins, 2 different types, and beta globins, the 3 different types here being shown, compared to the others. And essentially the point here is that they are very highly similar, more so than to other proteins known, and thus they all fit into the globin family. And they're quite different because they can be, I should say, quite different because they diverged so long ago, evolutionarily speaking, depending on how far back you go. The longer into history you look, the more potential mutations that would occur. Mutations happen, it's just a matter of giving them enough time to occur. There's also something here called exon duplication or exon shuffling, where one exon within a gene can be duplicated or added in on the homologous pair, and rearranging the order of the exons or the DNA within an exon, and thus altering the protein that's made in turn from those exons. So errors in meiotic recombination lead to the occasional mixing and matching of different exons, either within a gene or between 2 non-allelic genes, so adding in an extra exon. So sometimes these new sequences can be beneficial. They can make new proteins that provide new functions. That's evolutionarily advantageous. And sometimes, of course, they can cause problems or harm. And we're not just talking the proteins themselves, but of course, movements in all these different ways can also affect the pattern of expression and the regulation by messing with enhancers and so on, or even the promoter and what have you. And we're done.